right, so today we're going to be talking about the human immunodeficiency virus. Put virus down here. And as you can see, this is kind of a mouthful, so we call it HIV for short. And there's two super important things that you need to know about viruses. And the first one is that they are non-living particles, and they also need a cell to reproduce. And HIV is no different. It's just like any other virus. It's not alive and it needs a cell to reproduce. So the first thing we're going to do is we're going to take a look at what makes up the HIV virion. And the virion is just another name for a virus particle. So we'll write that right here. Virion. All right, so in the very middle of the virion, we have the HIV genome. And obviously it's not S-shaped, but that's how I'm going to draw it. And it is made of RNA. So we have an RNA genome. And surrounding the RNA genome and kind of protecting it, I'm going to draw it right there. And it is called the nucleocapsid. And it's just a protein coat that surrounds the genome. Inside a virus particle, we also have a bunch of proteins that are essential for the virus to function. And since this is not a virology video, we're not going to go into the details of what each of those little guys does. But they're in there. And surrounding this all is a protein layer called the capsid, which itself is surrounded by a lipid bilayer. And this lipid bilayer is just like one that you would find around a regular cell. And that's because it's actually stolen from a cell, because when HIV reproduces, it buds off of a normal cell. And that's something that we'll talk about a little bit more as the video goes on. So that means that there's, some random proteins up in here in the lipid membrane that were in the membrane of the original cell, but they don't really do much. What does do a lot is this protein complex, and it's got two important proteins in it called GP41 and GP120. And I'm not going to draw them in their correct conformation because that's not really important right now. But what you need to know is that GP41 is the one that spans the lipid bilayer. There's actually multiple copies of it, but I'm not going to draw them. And then GP120 is the bit that sticks out. And GP120 also has this little film of sugars on it. I'm going to draw that in. That's called glycosylation. I'm going to write that right here. Glycosylation. And GP120 really likes to stick to a protein called CD4. And I'm also not going to draw this one in its correct conformation because it's still not particularly important. But I'm going to draw it like this so that you can see that it sticks really well to GP120. And the special thing about CD4 is that it is only on two types of cells. So I'm going to draw them here. Here we go. They're on T helper cells and macrophages. And this video is not about what helper T cells and macrophages do, but just as a quick refresher, helper T cells ramp up the rest of the immune response. So they ramp up the B cells and the killer T cells to do their jobs. And macrophages eat anything that's foreign to the body, like a virus. So you'll have a helper T cell or a macrophage floating around, and then an HIV virus comes along, and it's got a GP120 on its surface, and the GP120 meets the CD4, and they dock together. And what happens next is that they fuse together. So we have a little virus bit over there, which is way bigger than it would be in real life. And here's the cell that it's fusing to. And when it fuses, it pushes all of its contents into the target cell. So we have our genomes with our nucleocapsid here. We have all of these pink proteins coming in. And now the cell's infected. And once the cell's infected, HIV can do something very, very special because it is a retrovirus. And that term's very important because it means that HIV can reverse the normal flow of genetic information in a cell, right? So normally we go from DNA to RNA to protein. And this is what's called the central dogma of molecular biology. Information flows from one to the next and you can't go backwards. But HIV can make DNA from RNA with the help of a protein, an enzyme, called reverse transcriptase, which is one of these pink little proteins that HIV spews into the cell when it fuses. So, when this happens, reverse transcriptase makes a DNA copy of the RNA genome and it just sticks itself wherever it wants in the host cell genome. And this means that the host cell thinks it's part of its own DNA and starts to make all of those proteins that those genes code for, which are actually the proteins that make up the capsid and the nucleocapsid and all of these other proteins that go in the virus particle. So what we end up with is this cell becomes a virus factory. It just gets filled up with these proteins. The proteins that go inside, and it gets filled up with capsid proteins. And what happens is that the virus particles assemble themselves into new viruses, new virions, and then a bunch of them all escape. So you get a ton of little virus particles. And then these guys can go on and infect a bunch of other innocent helper T cells and macrophages. So that's an overview of what the human immunodeficiency virus, or HIV, looks like and what it does. And in the next video, we're going to talk about how HIV causes the disease called AIDS.